All right. Hello, everybody. My name is Zhang, and I'm a new assistant professor in Computational Biology Department. And the goal of my research is to develop computational methods to decipher human disease using GWAS and functional genomics. So a bit of background, we know that the human genome consists of roughly 3 billion base pairs of nucleotides, and each of those nucleotides can be one of A or T or C and G. And those nucleotides can be different for different people, and there are around 10 million mutations or single nucleotide polymorphisms. For example, on this figure, the first mutation is from G to C. That means while a part of the population has a G on their genome, the other people in the population have C instead. So this difference in the people's genome is part of the reason that people are different and unique. And they may have different height, or they may carry different levels of risk for disease. And the goal of the research in our lab is to, first of all, identify the causal genetic variants for those phenotypes, and second, try to understand the underlying biological and molecular mechanisms for those genetic variants. So we design computational methods for analyzing large-scale genetic data, and one of these data sets is called genome-wide association studies. And this is this type of data that profiles the genotype data and phenotype data across a large population of people, and then they associate each of the 10 million SNPs or mutations to disease. And now we have those associations of the genetic variants to disease, then we can start trying to fill in what's in the middle between the genetics and disease in terms of molecular mechanisms. One way to think of those molecular mechanisms is in terms of the intermediary variables such as genes and proteins. And we know that genetic variants affect gene expression affect protein abundance, and then protein abundance manifest themselves in affecting the disease status of people. And also those mechanisms can be different for different cellular contexts, that in different tissues or organs or cell types, those relationships might be different. So how do we decipher this whole molecular system across genetic variants, genes, proteins, and across different cellular contexts? What kind of data do we have? First of all, like I mentioned, that we have the genome-wide association studies, which profiles the genotypes and phenotypes across millions of people. And those studies have identified tens of thousands of disease-associated SNPs. And second, large-scale consortiums have profiled a rich set of functional genomics data, such as chromatin accessibility, gene expression, protein abundance, etc. And those data are available across diverse cellular contexts, such as cell types or spatial locations. So with those data, in my opinion, the challenges are twofold. First of all, the statistical challenges are how to integrate different data types in a scalable and statistical principled way. And the biological challenges are how to systematically identify these genes, proteins, and cellular contexts. So my research is around addressing those challenges. For disease biology, I'm interested in the genetic basis of disease. First of all, I study the genetic architecture of disease, which is the study or the investigation of the distribution of the causal variants for a given disease. For example, what's the total proportion of the phenotypic variants that can be attributed to genetics, and which regions are more important than other regions on the genome, and what's the proportion of causal SNP for a given disease. And second, I try to identify disease critical cellular contexts. This is important because most of the heritable diseases and complex traits manifest in a very highly tissue or cell type specific way. For example, for schizophrenia, we usually think about brain cells, and for IBD or inflammatory bowel disease, it's usually immune cells. And identifying those disease critical cellular contexts is important for functional follow-ups and treatment development. And third, I also apply causal inference approaches to identify disease genes and proteins. Aside from the biological questions, we also look into the data collection process and try to optimize the experimental parameters to collect the data in a more cost-effective way. And I also design generic machine learning and statistical algorithms motivated by applications in computational biology. First of all, I design different data integration approaches to integrate different genetic and genomic data types. And second, because the data sizes are usually ultra large scale, which requires ultra fast machine learning algorithms. So we have a line of work of accelerating existing and popular machine learning algorithms to make them scalable to large scale data sets. So that's a brief overview of my research. Thank you for listening.